Hello, and welcome to the Jungle Histology online training series. My name is Ken Longenecker, and this is the first of 9 modules designed to teach anybody who is interested the methods my colleagues and I use for the rapid reproductive analysis of reef fishes. Now, our emphasis is on reef fishes, but you should be able to apply these methods to any type of fish. If you want more detailed information about these methods, you can look at any of the publications listed here. These publications describe the methods we will be teaching in this online series and show the results of rapid reproductive analysis for a suite of coral reef fish species. And you can download all of these publications and more from the Pacific Fishes website. Just go to the address listed here and click on the literature page. Before I get into the details of the methods, I'd like to explain why this site was developed. The simplest answer is that my colleagues and I hope that it will help with coral reef fish management and conservation. One of the simplest fishery management concepts to understand is to allow fish to grow to reproductive size before they are harvested. This should give each individual the chance to produce offspring or seed the next generation of fish. But unfortunately, we don't know that reproductive size for most fishes. Froese and Binland reported in 2000 that we don't know reproductive size for 83% of the fishes exploited worldwide. And that percentage is even higher for fishes living on coral reefs. So why do we know so little about these coral reef fishes? One part of the problem is the sheer diversity of coral reef fishes. About 25% of all known fish species live on coral reefs. And many people think that it would simply take too long and cost too much to study all of them. Another part of the problem is that many of the world's coral reefs are located in developing countries. And some people think these nations don't have the capacity to do the research or the infrastructure to support that research. So to address this lack of information on coral reef fishes and the perceived difficulty with working on them, we developed some methods for rapid reproductive analysis, which we lovingly call jungle histology. These methods are fast. Analysis of one species should take less than 2 weeks, and this should make that sheer diversity of reef fishes seem a little bit less daunting. The methods are also relatively cheap, and that should drive down the cost of analyzing all those reef fish species. It also makes these methods well suited for the developing nations where many of the world's coral reefs are located. All results are based on a detailed examination of reproductive tissue, and I'll go into the rationale for this in a later module, but for now I'd like to emphasize that these methods produce the sort of results that would be expected from a well-funded lab in any developed country. The methods use a bare minimum of equipment and supplies, so you can conduct your reproductive analysis right at the study site. This has a couple of advantages. First, you can work through your specimens, and if you find you need more individuals of a certain size, you'll still be in the field and will have the opportunity to collect them. The other advantage is that you can avoid the cost of transporting bulky specimens and avoid the sometimes onerous process of obtaining import and export permits for your specimens. These methods don't require a lot of the infrastructure associated with the laboratory in developed nations. For instance, you won't need electrical service, you don't need refrigeration. You can, and we have, analyzed a variety of species working in a hut in the middle of a jungle. Finally, I want to emphasize that these methods are easy to learn. This is not a staged photograph. Here we are working in a remote village in Papua New Guinea, and an 8-year-old boy is helping us to conduct our rapid reproductive analysis. It's our sincere hope that people viewing these online training modules will use these methods in their home countries. This concludes part 1 of the Jungle Histology training series. I'd like to close by thanking the Saltonstall Kennedy Grant Program for the funding to produce this module.